Okay, so hello everybody, I'm Jing. Uh, Today Jingle and I will be bringing you guys revision for chapter 6. So I will be focusing on the part 1 more. So without further ado, I will start with nucleotide. Nucleotide is made up of nucleotide is made up of phosphate, sugar, and nitrogenous base. So this is the structure of phosphate. Then sugar, there is two types. One is ribose that is used in RNA, and the second one is deoxyribose that is used in DNA. So how do we differentiate which one is ribose and deoxyribose? We need to look at the carbon number 2 of the ribose. It is bonded with a hydroxyl group, and the deoxyribose, it is bonded with only one H. So nitrogenous base, there is two types also. One is purine, which has two organic rings, and the second one is pyrimidine that has one organic ring. So the example for purine are adenine, guanine, and for pyrimidine, the example are thymine that is used in DNA, and uracil used in RNA, and cytosine. So all nitrogenous base will pair up with each other following a rule known as complementary base pairing rule, which a purine will always pair up with a pyrimidine. So a pyrimidine pair up with a purine by complementary base pairing, forming hydrogen bond. This is for DNA. And the same rule will apply for RNA molecule, where a thymine will be replaced by uracil to pair up with adenine by complementary base pairing, forming hydrogen bond. So the second one is also the cytosine, which is a pyrimidine, will pair up with guanine, which is a purine, by complementary base pairing, forming hydrogen bond. So now let's look at the question. Watson and Crick used a result from the work carried out by Erwin Chargaff. Erwin Chargaff is actually the ones who find out the rule of complementary base pairing. He found that the proportion of base A, T, C, and G were different in different species. But within each species, the proportion of A was equal to the proportion of T, means the ratio of A and T is the same. The proportion of G was equal to the proportion of C, means the ratio of G and C will be the same also. Will be equal. So name the base A, T, G, and C. So this is very easy. A is adenine, T is thymine, G is guanine, C is cytosine. So now let's move on to the Watson-Crick model, which describes the structure of DNA. DNA is made up of two long chains of anti-parallel nucleotides in which opposite complementary bases pair up by hydrogen bond. You can give example like pyrimidine will pair up with purine, like thymine will pair with adenine by complementary base pairing, forming hydrogen bond. And for the RNA, uracil will pair up with adenine by complementary base pairing, forming hydrogen bond to form ladder-like construction which twists into the shape of double helix. So when we need to describe the structure of DNA, these are the 6 keywords we need to include inside in order to score the full mark. Double strand, anti-parallel, complementary base, hydrogen bond, ladder-like construction, and double helix in shape. So suggest and explain how Chargaff findings help Watson and Crick work out the structure of DNA. This is the keyword of the question. So we need to include the 6 keyword inside to get the full mark. So complementary base pairing, the example of it, A with T, G with C, forming hydrogen bond, purine with pyrimidine, then 2 DNA strands, and the strands are anti-parallel. So Phoebus-Levin isolated the nucleotide of DNA and identified the carbohydrate component of each nucleotide. State the name of this carbohydrate component. So carbohydrate component is actually the sugar used in DNA, the keyword of the question. So just now I mentioned that sugar, there's two types. One is ribose, the second one is deoxyribose. So ribose will be used in RNA and deoxyribose will be used in DNA. So you guys can try this question. I will open to the floor. You guys can try what's the answer. Got marks one.
Yes, got marks.
Okay. So the first one is Yun, yes, is deoxyribose. So so carbohydrate component in DNA. Deoxyribose is used in DNA. So now I will talk about DNA replication. So before that, I will talk about the requirements for DNA replication. DNA replication requires DNA template, free DNA nucleotide, enzyme like helicase, DNA polymerase, DNA ligase, and one more is primase. So DNA template. Here is two polynucleotide strand. It is joined by the hydrogen bond between the complementary base. So there's two strands here. Each strand will act as a template. Then free DNA nucleotide. There is 4 different DNTP, which stands for deoxynucleoside triphosphate. So deoxy, we need to look at the sugar structure. The second carbon, there's only bonded with a H. So it is deoxy. So what is nucleoside? Nucleoside is actually the sugar and nitrogenous base component. So here is one nucleotide. The nucleoside is the sugar and nitrogenous base. This is known as the nucleoside. And triphosphate, tri means got three phosphate. So here got 3 phosphate. So why is there 2 extra phosphate? So there is 2 extra phosphate. It will be removed to release energy to join the two nucleotide to form phosphodiester bond. So this structure here is named as DATP. Why? Because you need to look at the nitrogenous base. Is adenine here, so it is A. If the nitrogenous base is guanine, it will be DGTP. If the nitrogenous base is cytosine, it will be DCTP. So basically, we're just changing the second alphabet. So the function of enzyme, I will talk about it later. So this is actually a summary of DNA replication. So the first thing is helicase will unwind the parental double helix. So what does actually helicase do? Helicase will actually break the hydrogen bond between the complementary base to form 2 straight chain using ATP. Then, by the way, there is two strands. One is the leading strand. The second one is lagging strand. So leading strand is the continuous replication, and the lagging strand will be the discontinuous replication. So the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. So what does DNA polymerase do? DNA polymerase will add a free DNA nucleotide with complementary base. Then hydrogen bond is formed to join the nucleotide together. Then phosphodiester bond will form. Then in the lagging strand, it synthesizes discontinuously the primase, which is the enzyme I mentioned earlier. It will synthesize a short RNA primer, which is extended by the DNA polymerase to form the Okazaki fragment. So here is the RNA primer, and this is the Okazaki fragment. So after that, the RNA primer will be replaced by the DNA, and then the DNA ligase will join the Okazaki fragment to the going strand. So ligase will actually join the 2 DNA fragments together to form phosphodiester bond. Then the phosphodiester bond will actually link the sugar phosphate backbone of both DNA fragments. So DNA will actually unwind the parental strand. Then hydrogen bond will break between it. Both strands will act as a template. The polymerase will actually add a free activated nucleotide, as I mentioned earlier. Then you need to describe it step by step. You can say after the DNA polymerase, then you can talk about the ligase will actually join the Okazaki fragment to the going strand. So here, replication bubble replication bubbles is actually the bubble is actually an open region of DNA where the DNA replication occurs. So bubble is actually created when the helicase separate two strands. So replication is semi-conservative. So semi-conservative is actually after DNA replication, the DNA double helix is made up of one new strand, which also can call as the daughter strand, and one old strand, which also can call as the parental strand. The one new strand is actually the replicated one, and the one old strand is actually the replication bubble is actually. Yeah, so this is the part of replication bubble.
What is the purpose of having many replication bubble in one DNA during DNA replication?
When the helicase will separate two strands, then it will have the replication bubble to separate it.
So there are many replication bubbles along one DNA molecule, right?
Yes.
So what is the purpose of having many replication bubble? Because this cannot be found in the bacterial DNA, which is circular, because bacterial DNA only got one site of starting point for DNA replication, whereas for eukaryotes, the DNA has got many sites of starting point for replication, and all of them will start with replication bubble. So that means there are many sites where DNA replication can start along the one DNA molecule. So why the difference? You can call someone and answer.
Oh, okay. I will call Andrea.
Is it because like so that like the DNA replication can occur at many points of the DNA strand simultaneously, so like you can replicate at a higher rate?
Yeah, so that you can complete the replication of the entire DNA that is very, very long in a short period of time, or else it will take a few days to complete the replication. So that's why our DNA is very long. So we need to have got a few points of starting point for replication at one time simultaneously, so that it will shorten the time that's needed to replicate the entire length of the DNA. This is a very important point that you all need to take note of. Okay, thank you, Andrea.
Okay, good question. And then, state the name of the part of the chromosome that prevent the loss of gene during DNA replication. So this is the chapter 5 one. The answer is telomere. Telomere is a region of repetitive nucleotide sequence at each end of chromosome that protect the chromosome from deterioration. So during DNA replication, the use of an incorrect base in the newly synthesized strand can lead to a mutation. So I will talk about a little of DNA substitution mutation. Actually, there is two types. One is transversion, the second is transition. So transversion is actually the interchange of purine and pyrimidine base. That means the two different types of base. And transition is actually the interchange of two-ring purine or the interchange of one-ring pyrimidine. So it is the interchange of the same type of nitrogenous base. So as you can see the picture here, transition is the interchange of the two-ring purine which is the A and G, the interchange of A and G, and the interchange. This is the interchange of the one ring pyrimidine. So one ring pyrimidine, the interchange of C and T, and transversion is the interchange of purine and pyrimidine, A, interchange of A and T, interchange of A and C, interchange of G and C, interchange of G and C. So a transversion event is where a pyrimidine is used in the newly synthesized strand instead of a purine, or the other way around. Name the two possible bases that could be used instead of cytosine in a transversion event. Just now I mentioned that transversion event is the interchange of purine and pyrimidine base. So you guys can try this question. Got marks also. Instead of cytosine. And transversion event is interchange of purine and pyrimidine. Cytosine is actually pyrimidine. Yes, guanine and one more. Yes. No. Uracil is pyrimidine. So
Yes,
yeah.
Sarah got it correct because she typed adenine and guanine, Sarah.
Yeah, okay. I'll move on to the next question. A transition event is where an incorrect purine is used or an incorrect pyrimidine is used. Suggest why transversion events are less likely to occur than transition events. So, as I mentioned earlier, transversion event is actually the interchange of purine and pyrimidine. And the transition event is just the interchange of the purine or the interchange of pyrimidine. So purine and pyrimidine are actually different size. 2 ring is for the purine and 1 ring is for pyrimidine. So it is a different size, and you can also explain it as purine is normally bond with pyrimidine to maintain the DNA double strand width. So if 2 purine or 2 pyrimidine bond together, it will distort the double helix width. Yeah, so that's the answer. Okay, that's all from me. I will pass it to Jing now. Thank you.
Thank you, Ying.
Now I'm going to continue with the chapter 6 revision. Figure 6.1 shows the formation of a polypeptide during translation in a eukaryotic cell. The first question, name the purine bases shown in figure 6.1. So as Jing mentioned just now, purine bases, we actually have 2. We have adenine and also guanine. So if we look at figure 6.1, we can already see that there is guanine and there is also adenine. So the answer is adenine and guanine.
Jing, can they write A and G instead of spell it out? Can they get mark if they only write A and G? You check the mark scheme.
The mark scheme actually,
cannot.
Yeah, you cannot write A and G. You have to write the full name, adenine and guanine.
Okay, thank you.
Okay, yeah, so let's move on to the next question. State the name given to the group of 3 bases found at J on the tRNA molecule. So at the diagram, you can see that this is the tRNA molecule. And we've already learned that for DNA, the term used is code. For mRNA, the term used is codon. And for tRNA, the term used is anticodon. So the answer is anticodon. And now identify the 3 bases at J. So does anyone want to try? I'll open it to the floor.
Yeah, Rebecca says anticodon, correct, 2 marks.
Yuhan CAG, and you are correct. The answer for the 3 bases is CAG. We look at the mark scheme here. Take a look at the mark scheme. You can actually also write cytosine, adenine, and also guanine. So for D, state how the 3 bases at J on tRNA interact with the bases on mRNA. I'm also going to open this question to the floor. Yes, Zisian, correct. Hydrogen bonds. So the mark scheme actually says that we should write hydrogen bonds form between complementary bases. As Jing mentioned just now, A, we can form complementary base with T or U in RNA, and G, we can form complementary base pairing with C.
I got question.
Okay.
For hydrogen bond, can I write H bond or I must spell out hydrogen?
You must spell out hydrogen bonds.
Okay, thank you.
Yeah. Okay, so let's look at the next question. The DNA in the nucleus is known as nuclear DNA. In the cells of the grasshopper, Tryptopus brunnius, 20% of the nucleotides in nuclear DNA contain thymine. Calculate the percentage of nucleotides in the nuclear DNA of Tryptopus brunnius that contain guanine, and explain your answer in terms of the structure of DNA. So we have to take note is that the question stated we have 20% of the nucleotides in nuclear DNA that contains thymine. And we have to calculate the percentage of guanine. So as we know, the ratio of A to T is a 1-to-1 ratio, and G to C is also a 1-to-1 ratio. So if for thymine we have 20%, and of course adenine, we will also have 20%. So we're gonna take 100%, we minus 40%, the 40% containing thymine and also adenine, and we get 60%. But please do take note that 60% is not the answer yet. We still have to halve the 60% to get 30%, because the 60% over here, it contains guanine, and it also contains cytosine. And the question asks us to calculate the percentage for only guanine, and that's why we have to halve the 60 to get the final answer, 30%. So for explanation, let's take a look at the mark scheme. So for just now, we got the answer 30%. So for explanation, we can say that bases are paired or are complementary. You can talk about the binding. Binding hydrogen bonds between A and T, C and G, or thymine or T pairs with adenine or A, cytosine or C pairs with guanine or G. We also can get marks for the calculation. So the calculation here, this is what I've already explained just now, and the answer is 30%. So moving on to the next question. This question is actually about translation. So I'm going to do a little recap about translation. So let's look at the diagram over here. Translation is the mechanism by which the sequence of bases in mRNA is converted to a sequence of amino acid to form a polypeptide chain. So as we know, translation, we actually have 3 stages. we have the initiation, we have elongation, when we also have termination. So let's look at initiation. So as you can see, this is the ribosome. So the ribosome actually binds to the first start codon, which is AUG, on the mRNA. We've learned that there is only one start codon, which is AUG. And two codons over here, the two codons on the mRNA are exposed in the ribosome. And the tRNA carrying methionine binds to the first triplet codon by complementary base pairing with its anticodon over here. And we've also learned that there is a P site and an A site. So what is P site? P site is the site where tRNA holds the polypeptide chain, and A site is where the tRNA brings in new amino acids. So let's look into elongation. Elongation, as we can see, the second triplet codon over here is exposed to tRNA. So the tRNA carrying the appropriate amino acid will bind into the A site over here by complementary base pairing between its anticodon and the codon on the mRNA. So when the amino acid, the first amino acid, methionine, and the second amino acid, they are side by side, there is this enzyme called the peptidyl transferase, and this enzyme actually catalyzes the formation of peptide bond between the two amino acids. So after the formation of the peptide bond, the tRNA in the P site over here, the tRNA in the P site actually leaves the ribosome to pick up another amino acid. And so the ribosome also moves along the mRNA for a distance of one triplet codon. So initially, the ribosome is over here. The ribosome is over here, and it moved along the mRNA for a distance of one triplet codon. And so the next triplet codon over here is now exposed to tRNA to bind in. And this process actually goes on and on. It repeats until the ribosome encounters a stop codon and there is no amino acid to bind to it. So the completed polypeptide chain then is released from the ribosome and the mRNA. So let's go back to the question. HIV protease is an enzyme composed of two identical polypeptide chains. Each polypeptide chain is 99 amino acids long. During translation, the amino acids are joined by peptide bonds to form the polypeptide chain. Describe how a polypeptide of HIV protease is produced by the process of translation. So let's look at the mark scheme. Firstly, we can talk about mRNA. It attaches to or associates with the ribosome. Two codons are exposed, A site and also the P site, or you can say that it's the first and second binding site. tRNA carries an amino acid to ribosome, or each type carries a specific amino acid, or first amino acid is the methionine, or tRNA with methionine. And the fourth point, anticodon on the tRNA binds to the codon on mRNA by complementary base and pairing between codon and anticodon, or hydrogen bonds formed between bases on codon and anticodon. So it says that it rejects the anticodon on mRNA because mRNA actually, the term for it is actually codon and not anticodon. Anticodon is for the tRNA. So for the fifth point, we can also talk about the start codon, which is AUG. Second tRNA with this amino acid binds next to first tRNA, and the two amino acids are held in place close to each other by tRNA binding. The seventh point, after the peptide bond formation, the first tRNA detaches and the ribosome moves along one codon. The process repeats or elongation occurs until a stop codon or polypeptide chain is synthesized. Next, tRNA molecules reused or leave ribosome to attach to another amino acid. You also get points if you stated that the enzyme peptidyltransferase is for the peptide bond formation. So let's take a look at this question over here. Table 2.1 shows two messenger RNA codons. Fill in the complementary transfer RNA anticodons in the spaces provided. So I'll open this to the floor. I'll open this question to the floor. CGC, yes, Zisian, you're correct. And also TGT, yes, you're correct. So let's look at this question over here. Calculate the minimum number of DNA nucleotides necessary to code for a polypeptide with 238 amino acids. So just now, I've already shown that this is the tRNA and it has anticodons, right?
Hang on. RNA cannot have T, right?
Sorry?
And the previous answer, the TGT, I'm quite curious because if it's RNA, it cannot have T.
Sorry. The answer is UGU.
Yeah, it's UGU.
This answer is UGU.
Who get the marks?
And it was Zisian just now. UGU is Andrea. Andrea,
thank you.
Sorry. Okay. Let's move on to the question over here. Okay, so this is the tRNA, and tRNA, we know that it binds to the codons on the mRNA. And the mRNA also forms, it binds to the code on the DNA by complementary base pairing. So this is the DNA, and this is the mRNA over here. And the tRNA, it carries one specific amino acid. So the question stated that there is 238 amino acids. So what we do is we get 238, we times 3. We times 3 to get the answer 714. And this will be the answer because the question is actually asking us to calculate the number of DNA nucleotides. So the answer is 714. Okay, so moving on. Figure 4.2 shows the first 7 amino acids of the beta chain of hemoglobin. The first amino acid in the sequence is valine, and the 21 base pairs in the sequence of DNA that code for these 7 amino acids. Table 4.1 shows the triplets of bases that code for 7 amino acids. Table 4.1 is over here. And using figure 4.2 and table 4.1, state what will happen to the sequence of amino acids in the first part of the beta chain of hemoglobin if the base pair at position 6 is deleted. So this is a question about gene mutation. So let's do a little recap about gene mutation. So as we know, we've learned that gene mutation, it can be caused by mistake during the DNA replication. It can also be caused by the DNA damages by mutagens, radiations, and also misrepair. And we've also learned that there's actually 5 types of gene mutation. So the 5 types of gene mutation, we have insertion, we have deletion, substitution, We also have inversion and also duplication. Yeah. So let's go back to this question. If the base pair at position 6 is deleted, so let's look at figure 4.2. So the sixth pair, the base pair at position 6 is actually G and C. So if both of these pair they are deleted, it's going to be GT, and the G over here will move over to be GTG, and over here is going to be ACT over here. And this part, CAC and TGA. So let's take a look at the mark scheme. So the mark scheme stated that the third triplet is a stop codon. It's a stop codon because we've already found that it's TGA. So let's refer to the table 4.1. So 4.1 is stated that TGA is a stop codon. So we know that the third triplet is a stop codon. Therefore, because the third triplet is a stop codon, there is only 2 amino acids that are joined by peptide bonds. So the chain has only 2 amino acids long. We still have valine and histidine as the first 2 amino acids. And because the third triplet is already a stop codon, therefore it's a very short molecule and the chain stops after histidine. Reject frame shift or description of frame shift because the question asks us what will happen to the sequence of amino acids in the first part of the beta chain of hemoglobin. So we have to talk about what actually happened with the sequence of amino acid. So we have to go into more detail. So let's look at the second question. If the 3 base pairs at position 7, 8, 9 are deleted. So let's look at the base pairs at position 7, 8, 9. Let me erase this. So 7, 8, 9, if they are deleted, we won't have leucine, right? So let's look at the mark scheme for the answer. A triplet is deleted. Therefore, the polypeptide has 1 less amino acid. And the polypeptide does not have leucine as the third amino acid as the triplet is deleted. Ignore leucine is not produced or made or synthesized. So the next question. The cell has mechanisms to repair the damage to DNA caused by hydrogen peroxide. Errors in repair may cause a change to the structure of DNA. Studies have investigated possible risk associated with foods and drinks that contain hydrogen peroxide. This is because the compound can be considered a mutagen. Mutagens cause mutation. So explain why hydrogen peroxide can be considered a mutagen. So just now I've already stated that gene mutation can occur due to DNA damages by mutagens, radiations, and misrepair. So let's look at the mark scheme. We get a point for hydrogen peroxide damage or breaks the DNA, and repair errors may occur. So hence incorrect nucleotide or base inserted during replication, or change in nucleotide or base, sequence of the DNA or RNA. Therefore, new allele may be formed and may result in an altered polypeptide. So the next question over here, table 5.1 shows the triplets of bases on the template polynucleotide of DNA for some amino acids. Figure 5.1 shows the base sequence in DNA and mRNA for the first 7 amino acids of the beta chain of hemoglobin. So for the first question, use table 5.1 to complete the figure 5.1. So does anyone want to try for the first one over here?
Yes, Andrea got it. ETG.
ETG. Yes, correct. So let's move on to the one over here. Does anyone want to try? Yeah, Johan got it. It's actually A, C, U. So for this beta chain over here, right? I'm going to, can you guys, this should be all right? So does anyone want to try? What's the amino acid over here? It's going to be valine. Valine is not the answer actually, it's actually leucine. Because for this, right, it's the DNA triplets. So the DNA triplets, we know that GAC is a leucine over here. So it's actually leucine. So let's take a look at this question. In sickle cell anemia, the amino acid at position 6 in the beta chain is valine and not glutamic acid. Explain how a single change in the DNA triplet for the 6th amino acid of the gene coding for the beta chain leads to the production of a different amino acid sequence. So let's look at the amino acid at position 6 of the beta chain. Let me erase this. So the 6th amino acid, it's a glutamic acid. So the question actually says that explain how a single change in the DNA triplet for the 6th amino acid can lead to a different amino acid sequence. So let's look at the mark scheme. So of course, mutation can occur and base substitution. So initially it was CTC. So if a single change, we change the T, we substitute the T to CAC. So the DNA has a 6 triplet, has CAC as the 6th triplet, and therefore the mRNA we have GUG as the 6th codon. And because the codon for the mRNA has changed, the tRNA, the anticodon on the tRNA also changes. So different tRNA involved, tRNA specific to valine and not glutamic acid. So the anticodon on tRNA has CAC. This is the anticodon. And tRNA brings incorrect amino acid or valine to ribosome. And therefore, incorrect amino acid incorporated into the growing polypeptide chain. So that's actually that was actually the last question. So that's all from me and Ying for chapter 6 revision.
Okay, thank you so much, Jing and also Ying, right, Ying? To Ying, right? Today's presentation is, I would say, I'll give you a 10 out of 10, right? Especially like your presentation, Jing is the pace of the presentation is just